part of our program with Martina McGloughlin.
Okay, thank you so much. Can you hear me? Can you understand my accent? Maybe. I actually grew up on a small farm a little bit east of here, like 6,000 miles east of here on the west coast of Ireland. So I'm actually familiar with being a farmer in addition to being a pointy-head academic, which I'm sure is the way many people view me. I'm now based at the University of California in Davis, and some of you I'm sure know that this is now ranked #1 in the world in food, ag and forestry research. So hopefully I can present a scientific perspective, because that's what I have been doing for the last 28 years. As Mary said, I'm not going to specifically address the particular measure here, because I wouldn't presume to know local politics. I wouldn't presume to know politics anywhere, to be quite honest with you. So I'm hopefully going to stick to the facts of science. And I really think, if we're all going to live in this world, the most important thing we need to do is to work together. You know, these two donkeys, while they're fighting, neither of them are going to eat. But once you start to cooperate, you can get access to all of the goods. And why do we need to do this? Again, as a scientist, I tend to look at big picture issues. So I'm often accused of being a reductionist, but actually I'm trying to look to the future, to the globe. We're going to have 9 billion mouths to feed by 2050. We are literally going to have to double our food supply to do that. And unless we find a whole new earth, we're going to have to use all of the technologies at our disposal to do this. The important thing is to know how to use these technologies together and in context to the betterment of everyone. Because to increase the yield and productivity of our crops, we're going to have to do this with a lot of disappearing resources. Our land is being degraded through over-cultivation. So cultivation looks pretty, it is not good for the soil. Our water supply is disappearing. If you lived in California right now, you'd know how much in demand water is. Mark Twain said, whiskey is for drinking and water is for fighting over, and we're really beginning to see that. And in Davis, I'll show you examples of how we're developing crops that are going to use way less water in the future. Likewise, we're trying to reduce the amount of inputs we use to grow these crops. We want to decrease the amount of chemical pesticides and chemical fertilizers that we need. And with the technologies that are being developed in Davis and elsewhere, and I'll give you some examples, we're going to be able to greatly reduce and hopefully over time eliminate the need for outside inputs to grow our crop plants. The other really important thing is to remember is we actually have been genetically modifying plants for about 10,000 years. And I've written a book about this. But don't worry, I'm not going to walk you through 10,000 years of research. But especially over the last 100 years, we've been using incredibly intrusive technologies to introduce traits into our crops. And most of these technologies can be grown using organic processes. Many of them would actually be banned if you're going to take a literal interpretation of the way your ordinance is read. So I'm only focusing on the science here, as I said, not on the politics. And I'll just give you some examples. So you heard about cross-hybridization. But if you look at some of these other technologies, for example, you heard about Japan, right? Being very skeptical about biotechnology. Well, I downloaded this picture from a Japanese site. This is irradiation mutagenesis. Right here, you have a cobalt-60 source that spews out about 90 becquerels of irradiation. It whacks a hell of a bang. It killed many of the crops. It doesn't have much effect on some other. And then some, you get a positive effect. And here's an example. This is an Asian pear. Now, Asian pears are notoriously susceptible to black spot disease. But using irradiation mutagenesis, you got a variety that's resistant here. Now, at the molecular level, they don't know what the hell they've done, because you are not, under any regulation, required to go in and say exactly what modification was done there. because these are generally regarded as safe. They don't have to be labeled, and you don't glow in the dark from eating them, I have to say, even though they have been irradiated. So you've either turned off a susceptibility gene or turned on a resistance gene, but nobody cares. The regulatory authorities don't care because they don't look at this. Likewise, spaghetti. All of the spaghetti you eat today is descended from varieties of durum wheat that was mutagenized in Italy after the Second World War in the nuclear research facilities there. And again, at the molecular level, you don't know what was done. Presumably, there was modification of the gliadins and gluteinins, which are the component of the proteins in this plant, in this particular wheat, that allows you to make good spaghetti that hangs together, so the good Italian spaghetti. And being Irish, of course, I have to talk about what we call here the juice of the barley, and that's beer. Now, all Irish beer, Scotch whisky and Irish whisky comes from a variety of barley that was modified in a nuclear research facility in England. You won't get any Scotsmen or Irishmen confessing to that, but that's the reality of it. Again, at the molecular level, who knows what was done? You're not required to go in there to see what type of modifications were done. All of these are generally regarded as safe. This is an example I actually took from Greenpeace. It says, if you eat asparagus, you will avoid eating GM foods. Now, do you know how asparagus is produced? Well, a very good way to select for recessive traits is to just cultivate the male part of the plant, the anther. Now, of course, you have haploid. It won't breed true. You actually then use a carcinogen called colchicine to modify the haploidy so you can get it to breed true thereafter. And they, in fact, are all called super males. So I have a feminist friend who won't eat asparagus anymore. That's a joke. But as I said, they can be grown organically, even though we've no clue at a molecular level what was done. So to me, coming forward, and I'm jumping ways here because I'm not talking about a bunch of these techniques, recombinant DNA technology is far more predictable, far more precise, and far more controlled than any of these older technologies where we didn't have a clue what on earth we were doing. We were just randomly mutagenizing and selecting or modifying in such a way that would give us the specific traits we were looking for. None of these are regulated at all whatsoever. Only the product of modern biotechnology is regulated. Now, going forward, we can use some really, really subtle modifications called genome editing that you can't even detect. So with these future techniques, how do you enforce that? Because you can introduce these modifications, you see the modifications, but you cannot tell that it was done using genetic engineering. So our ability to introduce these types of modification is becoming more and more nuanced as time goes by. And if you look at the difference here of what you're dealing with, using the older techniques, you're dealing with literally millions of base pairs there, and thousands of genes. With biotech, you're down to very few, and you have a much better notion of what they are because they're very well characterized. So you've probably been hearing about meta-analysis. It's meta-everything these days. With taking a meta-analysis, I actually call this meta-analysis the Sesame Street approach. Using all of these older technologies, right, which of those are more like their parents? The biotech one, taking the Sesame Street approach, the biotech one, where you've introduced a specific characteristic and you know exactly what you've done, is way more like its parents than any of the other techniques, and especially those that have been either chemically or irradiated to mutagenize them. There are huge variations occur at the genetic level. Using the most sensitive techniques that we have at UC Davis, transcriptomics, proteomics, metabolomics, economics, we can look and we can go in there and we can see what type of modifications have been done. And with biotech, you clearly see where the parents are. With these older techniques, you can't. Mm-hmm. And here is a very specific example actually from Davis. This gentleman here is Charlie Rick. He spent most of his life up the Andes. I'm being thrown out in 10 minutes. Now, Charlie had the widest collection of tomato germplasm in the world. And the processors came to us and said, okay, can you increase the soluble solids in your common or garden processing tomato, because if we increase the soluble solids, we have more bang for the buck, because you can't really sell water. Well, of course you can sell water in California, but water inside a tomato you don't get a lot of money for. So we went looking through Charlie's collection here, and this work was done by Alan Bennett, and they found this wild variety, like Aspersum chemaleski. You immediately can see it doesn't look very appealing. It's small, it's not very fertile, and given that tomatoes are members of the deadly nightshade family, in fact it's highly toxic. So the only characteristic we want in there is it's a sucrose accumulator. It's about double the soluble solids of your hexose. Now, it took many, many generations of backcrossing to end up with the phenotype we wanted. But we don't know how much of the metabolic pathways are still in there that codes for this toxin, the solanine. Using biotech, we didn't cross to any toxic plant. We just turned off one gene. We didn't introduce new genes, we turned off a gene. Now, going back to the Sesame Street approach, which of those is regulated and which isn't? It's actually the one that's crossed to the toxic plant, even though it's way more not like its parent. It still has the genes from the toxic plant, whereas the biotech one, you've just turned off a gene. But these are the biotech plant, where you turned off a gene, highly, highly regulated, to the extent that we can't afford to get it through the deregulation process, whereas the other plant, no requirement whatsoever from a regulatory perspective. So again, to say biotech crops, they're held to such a high standard. It takes 7 to 10 years of going through the process before they achieve what's called deregulated stage. There's at least 9 different review stages. And all of these older technologies that I told you, they're all assumed to be safe, generally regarded as safe, and are not required to go through any of this. One soybean alone was subject to 1,800 separate analysis steps before it was approved. These also include feeding studies, and if you look at dairy, beef, poultry, soybean, looking at dairy, beef, or poultry eating soybean and corn, they're equivalent in composition, digestibility, and feeding value to the non-GM variety, and all of this analysis has been done. The gold standard is substantial equivalence with the parents. And if there's any difference in there, then you go look and check to see if there's a problem with that difference. And nothing gets out unless it meets all of those health requirements and environmental and ecological impact requirements. And here's an example from our tomato. This is the tomato that's crossed with the toxic plant. This is the one where we turned off a gene. And there's piles more. That's just showing you an example of it. If we look going forward, the vast majority of the crops that are out there now are in the area of input or economic traits. Most are in what's called biotic stress. So it's all those other pests and disease and everything else that's competing with our food out there. It would be wonderful if we didn't have to control them, if we could do nothing. But if you do nothing, you won't have many crops, let me tell you. The area that's really becoming of greater importance is abiotic stress. As I just pointed out, in California, drought is a huge concern. So now we're seeing greater emphasis in the notion of developing these crops that are resistant to all of these environmental stresses: drought, heat, salinity, in some instances flooding, submergence, marginal soils. So there is a very big focus in the research that we're doing in Davis in these areas, and I'll give you some examples. And of course, consumers are hugely interested in the quality side, the output side. We're now starting to focus on the notion of not just improved nutrition, but looking at the functionality of food. And with biotech, we're getting an understanding of how food functions at all of these levels to supply more than nutrients to us, how we can optimize the quality of our life. So we're not running to the doctor to cure diseases, we're actually preventing them in the first place. And there's work going on the notion of using plants as factories, and of course you're very familiar, I'm sure, with plants potentially being used not just as biofuels, but as a renewable resource for all of the components in this room that we're now dependent on fossil fuels for. We can actually use plants to be the feedstocks to make all of these products here. The other important thing I think to remember from a biotech perspective worldwide is that this has been the most rapidly adopted technology ever by farmers all over the world. There's now over 430 million acres of biotech crops grown. And it's really interesting to me anyway, of the 18 million farmers that are growing these crops in 27 countries, 19 19 are emerging economies. These are small farmers, small holding farmers. I don't mean the smart farmers themselves are small. 90% of 16.5 million are resource poor small farmers, not big farmers in North America. And that's also really important. These are small farmers worldwide, and they see the value because farmers are smart people. They're not going to be spending money that's not value to them. If you look at the other specific advantages, and this is work that has just come out from Clive James, literally last month, he has determined that the value primarily to farmers worldwide is about $177 million. About 60% is due to reduced costs because you're using less chemicals to grow these crops, and because you are also increasing yield and you're reducing loss, you're reducing waste and loss. So 40% is about increased yield of about 377 million tons. The other side is we're reducing our pesticide footprint. It's down by about 1 billion pounds, and that's a serious amount. And that's active ingredient. That's not the whole amount that's sprayed out there. That's active ingredient. It's also allowed switch to conservation tillage. And really, when you look at conservation, when you look at tillage by hand, it looks pretty, but it is very, very destructive to the soil. If you can switch to this, and in fact this is called ugly farming. I know one of a farmer who says conservation tillage is ugly farming because of course you're leaving the debris on the fields, but it's a much, much healthier field. With this, you're actually reducing erosion because you're not ploughing into the topsoil, so you're reducing erosion by about 93%. The topsoil is not taken off and blown away, so it's preserved about 1 billion tons of topsoil. You have about 70% reduction in herbicide runoff, because remember, to control weeds, you have to use some complex cocktails of not very nice herbicides. Much better to switch to one that's not going to persist in the environment. You're also not driving back and over to control the weeds, so you have a reduction in fuel use of about 50%. And because, again, you're not ploughing in and you're not using all this fossil fuels, there's about a 60 billion pounds reduction in carbon dioxide emissions. And much of this is because the carbon dioxide is being sequestered in the soil. So this is the good sequestration, not what's going back in Washington. And this is about equivalent to taking almost 12 million cars off the road. The other thing is it allows what's called sustainable intensification. And an ecologist called Professor Green, very suitably, said some years ago that if we can produce more on less and leave more spaces wild, we'll have a far smaller impact on biodiversity than try to do ecological farming where it's not efficient and suddenly we're putting preserved lands, we're cutting down forests in order to grow sufficient crops to feed the earth. And that's a really important to remember that in context. The other thing that one of the other collateral effects of BT corn is a reduction in mycotoxins. Now this is actually a physical protection here. Here you have a child from Mauritius who has liver cancer, and you say, well, there's many reasons you might have liver cancer in Mauritius. In fact, when they biopsied it, they found what's called sphinganine lipids. Now that's indicative of a type of fungal toxin called fumonism. And this has been known to cause not just liver cancer, but liquefaction of animals' brains. It's horrible stuff. There's no good way to control it. But with BT corn, because you don't have the corn borer punching holes in the corn kernels, the spores can get in and the mycotoxin isn't produced, and you have a 90% reduction in this. This is estimated at a benefit of about $23 million annually, and this was done by an economist, Professor Wu. The other thing is, with BT, you only now have a very, very small part of one particular protein, a cry protein, that's absolutely targeted to the insect. You are not spraying with broad spectrum sprays. You're also not spraying with the full BT organism, which is mostly Bacillus cereus. And the parts of BT that are not good for you are the Bacillus cereus part, not the part that's introduced into corn, which is a small cry protein that's targeted to very, very specific pests. Another advantage of modification in maize is actually a variety that was approved in China, not here, some years ago. Now, one of the things that you actually see from a satellite, in addition to the Great Wall of China, is where you've got intensive production of monogastric animals. So monogastric animals are pigs and poultry, and us. We are monogastric animals as well. And the problem with why you see this type of eutrophication is you have to supplement their feed because they're not capable. Many of the nutrients in plants are not bioavailable. So you have to supplement their feed with organic phosphorus, and much of it ends up being secreted into the environment. Now, what the organic phosphorus is replacing is what's called phytic acid in the plant itself. This phytic acid is not only binding up the phosphorus, it is also what's called chelating divalent ions, so all of those minerals that are good for us. So you might remember Popeye the Sailor Man ads some years ago. Well, Popeye the Sailor Man was not getting as much iron out of spinach as they were telling you he was, because all that iron was bound up by phytic acid. So by having this phytase in there, this not alone breaks down the phytic acid and makes the phosphorus available, it also makes all these good minerals, all these divalent ions available as well. In addition, you don't have to supplement the feed of these animals, so they're not pooping it into the environment, so you're not getting eutrophication. So one enzyme, increasing the nutrition value and decreasing the pooping into the environment. So it's both healthy and environmentally sound. Now, I mentioned growing up in Ireland. This is actually something I saw growing up. This is a potato plot in Ireland, and in fact, this is copper sulfate. And copper sulfate is allowed, of course, in organic production systems because it's natural. But I think that's a false dichotomy. We should be looking at what's the best way to control pests and diseases. And it doesn't matter what source this comes from. Let's not making this false notion of natural versus unnatural. Let's look at beneficial versus not beneficial. And copper sulfate has a very high hazard rating by the EPA. It's considered relatively toxic, but it is acceptable to be used for use in organic production systems. And in a moment, I'll give you a much better answer than that. Here we have some grapes being aerial sprayed with malathion to control the glassy-winged sharpshooter. This is not a good thing to do either. This is not organic. This is clearly being used using a traditional pesticide, and I'll give you some examples of a better way to do this. This is an organic orchard, actually, in Washington, and it's been sprayed with antibiotics. Now, I don't know about you, but I would prefer not to have antibiotics sprayed in my apples, and indeed, we have a lot of alternatives to those. For example, I just showed you the potatoes there. Why do we spray the potatoes? Well, the reason we spray these potatoes is late blight disease is the reason that there are 40 million Americans claiming Irish ancestry. It caused the potato famine. It would be great if we didn't have to control these fungi, but this late blight fungi is responsible for 75% crop losses. Now, as I said, copper sulfate is one way to do it, but we actually have developed a potato that has 2 genes from a wild Mexican potato. These genes are from other potatoes. And the company that was developing them in Europe, this is BASF, have stopped even trying to do it because they got such opposition. I don't know about you. I would prefer not to spray with fungicide. I would prefer not to use copper sulfate. I would prefer to have these potato genes in there that potentially can lead to a $4.3 billion in savings. Savings from loss of the potato and savings from the use of fungicides or indeed copper sulfate to control it. The apple I showed you there. They're in fact apples that have already been modified to protect against fire blight, which is this bacterial disease that I showed you the antibiotics were being sprayed for. And they're called lytic peptide, but don't worry, there won't be a pop quiz afterwards, so you don't need to remember the name. But there are apples out there that are protected against this bacteria, and you don't have to spray with antibiotics. Likewise, there are scabs on apples that are caused by fungi. And fungi, like insects, have chitin in it. So they have introduced chitinases, indo and exokitinases, into these Macintosh trees, and they increase resistance against these scab fungi. I mentioned about the grapes that are being affected by Pierce's disease. Well, Pierce's disease is caused by a bacterium again, Xylellum fastidiosum, and this particular bacteria is introduced into the grapes by a glassy-winged sharpshooter. And right now they can only control the vector, so you spray the vector. Not nice to have all of these insecticides being sprayed down on you. Well, in fact, taking 2 genes from spinach, you can get resistance against this bacterium. The other advantage, of course, if you're trying to control a vector, it's not the perfect way to do it because all it takes is 1 prick and that's the end of it. So with this, you have internal protection. And again, it's natural genes from spinach and you get protection, and definitely preferable to than spraying malathion. Those of you that like orange juice in the morning, well, that will disappear one of these days because it's been destroyed by another bacterium that causes citrus greening. There is literally no way to control that right now. What they're doing in Florida is ripping out the orchards and burning them. They are using so many pesticides, they are killing the bees, the very bees they need to pollinate these trees. Again, there is a biotech solution. In fact, there is only a biotech solution to protect against citrus greening. And you may have heard about the papaya in Hawaii. My good friend, Dennis Gonzalez, he actually got his PhD in Davis, but he did most of this work in Cornell, and now he's back in Hawaii, where he comes from. The papaya were being destroyed by a virus called papaya ring spot virus. And there's no natural resistance against this virus. It doesn't matter how good a plant breeder you are. If the genes aren't there, you are not going to get them in. And he noticed that the varieties that were infected with a mild version of the virus was actually protected against the more virulent strains. So it was almost like these papayas were being vaccinated. And that's in fact quite similar to what happens to you and I. When you get vaccinated, you produce antibodies that then protect you later on. Now, of course, plants don't have antibodies, but they do have this system called RNA interference. And so, in fact, it works at the RNA level. It doesn't even have to be translated into a protein. It was once known as post-transcriptional gene silencing. Again, no quiz, so you don't need to remember this. The other really big advantage of this, when he introduced it into the particular variety he first succeeded in getting the gene in there, it didn't have the right flesh color. So he crossed it with a variety, it's called Sunup, to give them the nice orange color they wanted. And he now also got hybrid vigor. So he increased production in addition to increasing resistance. The other thing it's doing is organic farmers actually are quite happy about this because it's removing the viral reservoir. It's like herd immunity. It's getting rid of the reservoir, so you're reducing the impact on the organic farmers themselves. The same thing has been done for grapes. For plums. So all of these may be disappearing if we don't use organic biotech approaches. I had mentioned about environmental stresses as we're looking going forward to increasing drought, salt, heat, flooding, nutrient efficiency. With biotechnology, we actually can introduce various types of genes, and there's a bunch of different sort of what's called master switches, like transcription factors, or the genome editing I told you about, that will allow us to decrease yield loss under severe drought conditions. One particular one, there was a 30% yield advantage compared to conventional hybrids under drought conditions. One of my favorites is from Davis, and I call this the resurrection gene. Both of these crops were subjected to 15 days of drought. This is the control, this is our biotech one. When then you re-water them for 7 days, the control are still dead as a doornail, but our biotech variety with this resurrection gene popped back to life. So again, if you're looking at drought tolerance, this is the type of thing you want. The other really big problem we have, of course, and one of the side effects of irrigation is salination. So irrigation is great, but the water evaporates and the salt stays in the soil. And in fact, 20% of irrigated land worldwide is no longer arable because of the degree of salination. And in Davis, again, we've taken this gene that basically acts like a pump, and it takes the salt and sticks it in a vacuole. And these are plants now grow and thrive in irrigation water that's 50 times saltier than saltier than normal. 1/3 as salty as seawater. So we can put all that land back into production again. You hear, of course, that we use way too much fertilizer. Well, in addition to carbon dioxide, nitrous oxide, in fact, is probably an even worse greenhouse gas that causes increase in temperatures. Now, using this particular gene here, it's a nitrogen use efficiency gene. When you introduce this into a plant, you're going to get the same amount as with conventional plants, but you're using 30% less nitrogen. Now, nitrates are pretty expensive, so you're saving the farmer, but more importantly, you're saving the environment and you're saving the air. So 3 savings there for 1 gene. I mentioned that nutritional characteristics are something that people are really interested in as well. And here's something that's been done by your neighbor in Idaho. So when you cook potatoes, this asparagine here can result in the production of acrylamide. And acrylamide is a carcinogen. Not a really bad one, but still much prefer not to have those in my fried potatoes. With this particular, using the same RNAi, they call it in 8 genes, the same approach again, you can reduce this by about 80% less acrylamide. Likewise, taking this RNAi approach against another gene, you can reduce black spot from bruising. So you don't need to throw away or discard as many potatoes. So you're saving waste there. And another version is being used to modify the sugars so you get a nice golden color. And again, you're not throwing away potatoes because you're getting the wrong color. You can cook those in soybean oil that has been modified, this particular second one here, it's modified so you get high oleic, low linolenic acid. And this is basically a highly stable soybean oil. In addition, you don't have to hydrogenate it, so you don't introduce trans fatty acids. So it is far healthier, it is low saturated, and it's very, very stable. So when you cook it, you're not breaking it down and you don't have to throw it away as much. So you can cook these highly safe, high quality potatoes in this high quality soybean oil, and you have much better oil and much safer oil and much healthier potatoes. On the other side here, we all hear of the importance of omega-3 fatty acids, and omega-3 is important for cardiovascular health, for our mental faculties. In fact, 30% of your brain is made up of docosahexaenoic acid. So if someone calls you a fathead, that's actually a compliment, because you need these fats to think. Right now, we're dependent on fatty fish as our main source of omega-3 fatty acids. But by taking a gene from algae and putting it into soybeans, 1 acre of soybeans can replace 10,000 salmon. So you can protect your coho salmon taking this approach. You may have heard of golden rice. Well, in areas where rice is the primary source of nourishment, primary source of calories, vitamin A deficiency is prevalent. And vitamin A, it's a very essential multifunctional nutrient. The one most of us see, of course, that it's critical for vision. And with this vitamin A deficiency, you do end up with macular degeneration and ultimately blindness. But it also plays a very significant role in the immune system, and it's crucial for growth and development. This is primarily, as you can see, a disease of the poor. It kills between 2 to 2.8 million people annually. These are mostly children under 5 and women. Golden rice has genes in there for beta-carotene, which is a precursor for vitamin A. Our body turns this into vitamin A when you digest it. And you only need about 40 grams a day to offset the deficiencies. You're not going to make you super healthy, but it will offset the deficiencies. It's far cheaper, it's easier to distribute, and it's more sustainable than all of the other sources of vitamin A out there. An estimate was just done, and this paper just came out last month from David Zilberman in Berkeley. Since 2002, he estimates that the delay in application of golden rice in India alone has cost 1.4 million life years. And this is an economist that did this. This isn't a scientist or any of the people that actually did the work on vitamin A. There's a bunch of other work that I don't have time to go into, where we're looking at decreasing allergens, decreasing toxins, and increasing high quality nutrients in our crop food. But of course, all of this is still sitting in labs because of the huge cost of going through deregulation. And just to briefly mention the notion of good farmers, here is a very specific example from Idaho. This didn't come out here, sorry. This is Fred Yoder and his neighbour. He grows biotech corn, his neighbour grows organic corn. And it's very easy to see if cross-contamination is occurring, because biotech corn is yellow and the organic corn has a blue outer layer. And they've been doing this for about 10 years. And what they do is they delay planting. So you stagger the actual planting time. So when one plant is actually receptive to pollen, the pollen is not being produced in the other one. So it's really, the cooperation is hugely, hugely important. And many crops, this is true, many crops containing diverse market types do coexist. Sweet corn and field corn production, oil and confectionery sunseed production, that's done all over California, canola and rapeseed. These types of diverse production of various seeds have gone on for a long time. But zero tolerance is extremely difficult to attain. Coexistence is not possible without pragmatic proportional thresholds. So this notion that exists, and I don't even understand why it exists in organic production to say zero tolerance, when you do allow up to 5% of contaminants from other sources, it seems to me absolutely ridiculous. But in the seed industries, the producer who is looking for the high purity for a high value market, they need to meet market expectations. And you cannot legally downgrow or decertify growers if there is unintentional presence. No producer has been impacted for unintentional presence, and it's really important to remember that. The other thing it's really important to remember is these products have been studied by scientists, research organizations, health and food safety organizations, and regulatory organizations all over the world. World Health Organization, the American Medical Association, and they said, well, this is what the World Health Organization said, GMOs offer the potential for increased productivity, improved nutritional values that can directly contribute to enhancing human health. About 2 trillion meals with GM ingredients have been eaten around the world without a single substantiated case of ill health. And there's now been over 700 peer-reviewed scientific studies. This is done by scientists. This is done by the experts. It's reviewed by the experts. They've all concluded that these are safe. And if you look at one of the areas, again, that's highly skeptical, I started with Japan, I'll finish with the EU since that's where I'm from. They looked at 25 years of research. They looked at 130 research projects with 500 research groups over 25 years. And they said, there is no scientific evidence associating GMOs with higher risk for the environment or food and feed safety than conventional plants and organisms. They went on to say, because we have such a better notion of what we're doing at the molecular level, and because they are so highly regulated compared to all of those other techniques, they're probably safer than many conventional methods that we've used to introduce crop plants into our traits into our crop plants. And that really, it's not about science anyway, but I am about science. So just again, to list, as Brian did, these are the scientific or medical bodies that have an opinion on the safety of GMOs. These are the ones that are generally positive, and like Brian, I could go on for 40 or 50 pages, and these are the ones that are generally negative. There are none that are generally negative. Everyone will say you need to continue to look and make sure that if there are concerns that they're addressed, but they all are positive at this point in time. And here's an example of one, I lied when I said there wouldn't be a prop quiz. So if I was to say this product contains potentially toxic genes from the deadly nightshade plant, where do you think this label should be? Potatoes and tomatoes, exactly. In the interest of accuracy, which product should carry this label? Potatoes and tomatoes. Okay, let's see if we're going to label with genetically modified cookies here that have genetically modified ingredients. So, produced with genetically engineered ingredients. What does this tell you? Okay, let's look at everything else we should put on the label. So we're being scientifically accurate here now. Product of irradiation breeding, derived from chemically induced mutations, product of Y genetic cross and ovule rescue, constituents altered by transposon mutagenesis. What should that be on?
Spaghetti.
Pretty much everything, yes. Now, would the consumer find this informative? I think the consumer would find this scary as heck. Yes, your spaghetti, your asparagus, your pears, and I could go on and on and on. Grapefruit have been modified using some of these techniques. and only very, very few crops in comparison to all these other crops have been modified using the products of modern genetic engineering. So oftentimes, the risks that people are scared of are not the risks that are actually the greatest hazard to them. When you feel you're in control of a particular situation, you actually consider that it's less risky, where in fact it could be far more risky to be chowing down on the older, and this isn't the genetically modified one, the older Frito-Lays cooked in oil compared to our modern reduced acrylamide, reduced saturated fat, genetically modified oils and potatoes. And with that, I will leave it. Thank you. Thank you.
Anybody have any questions for her?
Okay, wherever. Yeah, we can start with you, Davis.
Go back to your earlier slide that said benefits to date.
Repeat that, please.
He wants me to go back to benefits. Sorry, this takes a while because they're all.
What time period are you talking about?
So this is looking. So this here is looking from the introduction. So 1996 to date. Yeah.
Not as good as at arithmetic that I used to be. But starting at the top, since 1996, there has been a total
Yeah, this is this
77 million more tons of food.
increased food productivity.
Okay. If you would allow a number, say, close to 400 million acres worldwide, and $177 million, if I've got this right, and I may not, so let's talk about it, it's like $0.50 per acre.
Oh no, but the fact is you're producing this on way less land as well. So it's really important to look at the context here.
Tell me first if I did this
So
right.
if you could stand up and say your question.
Addition on top, not total.
No.
Yeah.
Total. You have $177
okay.
million of added benefit.
Could you stand up and so we can hear your question, sir?
Oh my God. Okay.
Thank you.
I tried to figure out the arithmetic on benefits today. And it shows $177 million added.
Added value on top of what you would have gotten.
Okay. So when you divide that by 400 million acres worldwide of GMO crops, you get added value of 50 cents per year.
Yeah, but what you're not taking into consideration is the reduction on the other side. So that's why I presented it as all together. The reduction in pesticides, what I'm talking about is the actual physical reduction of the amount of chemicals that we use to grow these crops as well. You have to look at the whole package, not just specific numbers.
Okay, it doesn't say that.
Oh, it does. Here.
You have to look at the benefits from the use of lower amounts of chemicals.
Yeah.
But the USDA just told us in a report that it's not been fewer chemicals. Roundup has gone up 10 or 11-fold.
No,
compare.
but tell me how you grow plants without control. It's not Roundup going up, it's everything else that's disappeared, that's gone. We're not using these complex cocktails that hurt the plants themselves, that persist in the environment. In fact, I know a farmer who said for the first time ever he took a vacation because he is spending less bum time on his tractor, is how he put it actually. And he's not spending all this time mixing up complex cocktails that persist in the environment, that damage the very plants that you are trying to protect, in addition to getting rid of the weeds. So it's a whole complex.
The USDA numbers say you have 90,000 tons of Roundup being used.
But what are you displacing? And that's what it's really important. I'm talking about pesticides as a whole. I am not talking about a particular pesticide. And in fact, I specifically said that. You are switching from one to another under conservation tillage. But the absolute amount on larger terms is a reduction and a switch to a type of chemical that doesn't persist in the environment, that is healthier. It will be wonderful, and going forward, if we can eliminate this altogether, but you have to control pests and disease and weeds, and using biotech approaches, this is more sustainable.
On the yield, then, that's over since 1996.
Yeah.
17 years. 377 million more tons than we would have, divided by 400 million acres, it's one.
No, but it's not 400 million acres. That's 400 million acres this year. We started
It
with
was only like 380
nothing.
the year before, and 300.
It's going down over time. So you're actually looking at the cumulative over time.
There's other questions.
Yeah, sorry, go ahead.
Now, would you hang on to that question, maybe talk with
Yeah,
Martina a little later? We
Sure.
get some more
Yeah.
questions.
I didn't believe that was from Forbes.
Mm.
Could you stand up?
Can you stand up and speak louder?
Sorry about that. So I believe that it was from Forbes actually that the
Not
information
USDA.
had come about it going up tenfold, and
Actually, there's a graph published by the U.S. Department of Agriculture that was published in Forbes.
that was the source of it. Okay, I didn't understand. But one thing I did want, if you have a moment to go over, something that's really important that I believe that the audience should be aware of is this idea of phytoremediation and hyperaccumulation of toxic elements in the soil. You can use plants to essentially.
Yes, I will.
This is something that you can use biotechnology to naturally make a remedy for the soil.
And I have a whole chapter in my book about that. I didn't go into it here because I only had a certain period of time. But you can use plants to clean up the soil. In fact, we're developing plants in Davis, and it has been done elsewhere, that will take toxins and heavy metals from the soil. Yeah.
About two of the things that I noticed I had a question was your comment about BT and how that improves things because the protein is targeted to very specific.
Lepidoptera.
Okay, if you reference Stephanie Senna, the MIT.
Oh, she is an artificial intelligence person. She has no biological training whatsoever. And I've actually looked at her papers, and she does this correlation with causation. In fact, some of the inferences she makes, you could say, looking at the increase of cell phones will potentially increase other health problems. The increase in organic food consumption, you could potentially say you're having the same increase in gluten intolerance, I think is one of the examples she gives. You cannot have correlation and causation unless you can absolutely prove the relationship between the two, and she does not do that. She just throws numbers up there.
The other one is on the rice, the golden rice. That was on the cover of Time magazine. It was supposed to save all these children. Well, I've heard since then, Vandana Shiva has blown holes.
No, she actually hasn't. In fact, this work that was done by Ingo Patrikas, who I know well, the reason that it isn't out there saving people is because it still hasn't been approved. All of these theoretical risks.
So what's the amount of grams?
40 grams provides a sufficient amount of beta carotene that's converted by our body into vitamin A per day.
How much rice would you need to eat?
That's less than half a bowl of rice. That is a tiny amount of rice that will meet this requirement. And this was done by researchers in Cornell that actually did this assessment. And if you talk to Ingo Patrikas, he says he feels those that have prevented this being developed, and especially prevented it being deregulated, are responsible for all of these deaths. And in addition, this includes blindness in addition to actual deaths, developmental disorders. And last year in the Philippines, the very trials that were being used to look at the potential impacts from a health and environmental perspective were pulled up by activists. They thought they were by farmers in the Philippines, and the farmers said, we wouldn't touch this. This is really important to us from a religious perspective. We would never pull up rice. They found there were activists that had been shipped in, and when they traced the money back, it was to the Norwegian government. I mean, it was the strangest correlation I had ever seen. And these were the trials, yeah.
Let other people ask questions.
Why don't we have this?
Because of the fact it's been held up for theoretical problems that don't exist. So I actually think, personally, I think this is a disgrace that this isn't out there, in my opinion.
How long back
One
here?
back there, and then one over
can you comment on the level of total agricultural production in these counties that have introduced GMO laws in the state of California?
You mean the anti-GMO laws?
Yeah,
Well, many of them, many of them don't grow the commodities that have been deregulated at this point in time. And there's very few actual, for example, whole fruits that are, that would be specifically grown in those counties. Have gone through the deregulation process at this point in time. So I cannot tell you the specific impact on that, but they are not some of they would not be the counties where you would have large commodities. The other interesting thing about that, if you look at the way some of those are defined, for example, Mendocino, it refers to DNA as a complex protein. So if any scientists choose to challenge it, this law would not hold up in court because, of course, DNA is not a complex protein. The other really interesting thing is almost all of your enzymes, the enzymes that you use to make cheese. So any of you that like to eat cheese, almost 100% of your cheese is made with genetically modified chymosin that has a gene from the forestomach of an unweaned calf, and it is not it is not labeled, or in fact nobody has an issue with it, including all of France. Over 80% of all of French cheese is made using these genetically modified chymosin. Likewise, the enzymes that are used in your fruit juices, in your toothpaste, in your detergent, all of those, a large portion of those, are now genetically modified, but because they're considered part of the processing, they're used everywhere all over the world with no labeling or no actual push to have them labeled either. So just another point that's often forgotten. Oh yeah, okay.
Well, I just actually have two questions. One is, this lady was asking about the golden rice. How long did it take for this to be developed, and then why is it?
So it was ready in 2002. All of the process, all of the regulatory oversight assessments with respect to food safety, the efficacy of the product itself, and also looking at potential environmental impact. All of this was achieved in 2002.
Yeah, that's not what I asked. How long did it take to develop that?
Oh, it was developed over about a 15-year period, I guess, when they first started to do this work.
How much money did that cost?
So much of this work was done in Zurich, actually in ITEH, and most of it was from grants from the government of Switzerland, so I don't know how much that was.
So the taxpayers paid for that for 15 years, is that what you're saying?
Yeah, so
and it hasn't been released yet?
Because of the process of going through deregulation. So it should be out there. There is no issue with it.
So could the government have just given them vitamin A pills?
The problem is distribution. And this you can have forever. If you teach, it's sort of the same as, give a man a fish, you feed him for a day. Teach him how to fish, you feed him for a lifetime. Same here.
Yeah,
it's packaged technology in the seed.
I didn't actually think most rice had vitamin A in it.
Actually, rice does have vitamin A. It has it in the vegetative portion. It's just not produced in the part we eat.
Exactly, that was my point. So my other question, actually, so thank you for that, because I wanted to follow up on Blanche's question. The other question I had, I know that BT corn is grown, and so what I'd like to know is, since the USDA has registered BT corn as a pesticide, the reason is because BT works, as most farmers know, by exploding the stomach of insects. That's how the insects are killed. And of course now the BT corn is being fed to animals and maybe in people.
But in fact it is far safer than sprayed BT, which is essentially Bacillus cereus, and Bacillus cereus is far more toxic, and that's allowed in organic production systems. The whole bacteria. Here you just have a proportion. Yeah, sorry, go ahead.
That's my question. So I know that the organic BT is washed off and it disappears in the sunlight. So my question is, now that the corn rootworms are becoming immune or being able to tolerate the BT corn that they're eating, I believe I've heard stories that there is BT, the super, I guess I should call it super bugs, just like we were talking about super weeds that are occurring. And so as this genetic engineering continues to happen, it seems like
No, this is biology. And so what you do is you use genomics to increase the variety. So you can have multiple, many versions through selected evolutionary processes using genomic approaches. And with biological systems, nothing is ever perfect. So you use basically what you do, rather than chemicals, you use gene rotation and pyramiding. And each gene works in a slightly different way, but it's at the genetic level. It's not spraying chemicals outside.
So I think they're going to go to 2,4-D.
That is nothing to do. Sorry. It's BT I was talking about. Yeah, sorry.
One more question. I think there was
Yeah.
one over here somewhere.
Well, I wanted you to address the super weed question.
Yeah, and that's the other thing. You have to manage it, and that's an important component that's always looked at to determine if there is a potential there. Because you also need to remember that one gene does not a weed make. That when you transfer, when genes are transferred or when genes flow, it's taking half of the genetic material of the domesticated plant, which is going to have a less capability of surviving in the wild if you don't provide selection pressure. Using older technologies where you also had introduction of these both, for example, herbicide tolerance such as Clearfield, they're not controlled at all. You're not required to go through any of that, and you have a much greater potential for development of superweeds. But you have over 300 other chemistries to deal with it. Management is hugely important. Biotech is not a panacea for all the world's ills. Proper management of your land is massively important component thereof.
I came tonight to be a critic of what the doctor here was going to talk about, and my wife having manipulated barley with colchicine at both UC Davis and Colorado State University, looking for mutants in barley that might be less susceptible to certain diseases, was really a very crude.
It was very crude, very crude.
That they did make some progress. In fact, my colleagues at Davis, Charlie Schauer, at one point during his review process, another colleague, Dr. R.S. Loomis, put a comment on that the amount of improvements that Charlie made using those old crude methods in improving barley would have paid for the agronomy department at Davis for when it started. As far as the tax increase value of the improved crop production. I think she did a very nice job tonight, but I've been up down here for 20 years, shopping in Medford, living in Fort Jones over the border, come up here because food's cheaper, and there's more stores here. But I've become appalled in the last 20 years with the copywriting of professional research. When I was a professor in publishing, we paid page charges to get our research published.
We still do.
And today that we have entrepreneurial journals that are copywriting all the information. So now I'm here on a nice big iMac computer, and if I want to read the publications and delve more into Roundup residues and things, I got to pay for the damn stuff.
That's why we're pushing to have them all free.
So what I would ask our guest tonight, would she form an effort at Davis
yes, we are.
to reinitialize making our taxpayer-funded research available to everybody on the internet? And that's number one request. I'd like to have a way that the published literature can be read by everybody, since everybody in the United States paid for it.
Yeah.
We shouldn't have to be paying these journals anymore to be able to get the average is about $30 for a 6 or 7 page technical article with all the figures and tables. Ridiculous. How can I, as a scientist up here, research anything with those kind of costs? So my other item is, though, is I have a colleague a farmer in Scott Valley who actually graduated from UC Davis. He's an organic farmer now, and he's concerned about Roundup residue. I would like to see the program at Davis do a thorough assessment of the residue in soils, particularly in the Midwest, where they've been used the longest with BT corn or cotton, and let's see a solid review of buildup residue effects, because my colleague right in Scott Valley feels like we're so close to organic in the whole valley, it's really, and I did talk to our farm advisor this morning, our alfalfa farm advisor, who's the best one in the state of California, Steve Orwell. He thinks we got 1,000 acres of Roundup alfalfa. And that's very disturbing to some of our organic people in Scott Valley. But right now, we can't even get the literature. So I'd like to see a review of
and in fact, Jonathan Eisen has started that very process in Davis to make literature available for free if taxpayers have paid for it. So Davis is actually one of the people pushing that whole thing, Jonathan Eisen.
I would greatly appreciate
and
that.
So he's making that free.
And you're actually in a key position. Because I do believe genetic engineering will feed the world. It's the only way.
And the other important thing to remember with glyphosate tolerant, it breaks down, the enzyme breaks down the glyphosate. There is less glyphosate on the plant because it's broken down by the very enzyme. So, sorry.
Thank you for making that a quick question.
Oh, really good. Yeah.
Thank you,
Thank
Dr.
you.
McGloughlin.